Let's continue from the previous lesson about central nervous system. So, characteristics of some important small molecule transmitters. Acetylcholine is secreted by neurons in many areas of the nervous system, but specifically by the terminals of the large pyramidal cells from the motor cortex. Several different types of neurons in the basal ganglia. The motor neurons that innervate the skeletal muscle. The ganglionic neurons of the autonomic nervous system. The postganglionic neurons of the parasympathetic nervous system, and some of the postganglionic neurons of the sympathetic nervous system. In most instances, acetylcholine has an excitatory effect. However, it is known to have inhibitory effects at some peripheral parasympathetic nerve endings, such as inhibition of the heart by the vagus nerve, and norepinephrine is secreted by the terminals of some neurons whose cell bodies are located in the brainstem and hypothalamus. Specifically, norepinephrine secreting neurons located in the locus ceruleus in the pons send nerve fibers to widespread areas of the brain to help control overall activity and mood of the mind, such as increasing the level of wakefulness. In most of these areas, norepinephrine probably activates excitatory receptors, but in a few areas, it activates inhibitory receptors instead. Norepinephrine is also secreted by most postganglionic neurons of the sympathetic nervous system, where it excites some organs but inhibits others. Then dopamine is secreted by neurons that originate in the substantia nigra. The termination of these neurons is mainly in the striatal region of the basal ganglia. The effect of dopamine is usually inhibition. Then glycine is secreted mainly at synapses in the spinal cord. It is believed to always act as an inhibitory transmitter. Then GABA is secreted by nerves terminals in the spinal cord, cerebellum, basal ganglia, and many areas of the cortex. It is believed always to cause inhibition. Then glutamate is secreted by the postsynaptic terminals in many of the sensory pathway entering the central nervous system, as well as in many areas of the cerebral cortex. It probably always causes excitation. Then serotonin is secreted by nuclei that originate in the median raphe of the brainstem and project to many brain and spinal cord areas, especially to the dorsal horns of the spinal cord and to the hypothalamus. Serotonin acts as an inhibitor of pain pathways in the cord, and an inhibitor action in the higher regions of the nervous system is believed to help control the mood of the person, perhaps even to cause sleep. Then nitric oxide is especially secreted by nerve terminals in areas of the brain responsible for long-term behavior and memory. Therefore, this transmitter system might in the future explain some behavior and memory functions that thus far have defied understanding. Nitric oxide is different from other small molecule transmitters in its mechanism of formation in the presynaptic terminal and in its actions on the postsynaptic neuron. It is not performed and stored in vesicles in the presynaptic terminals as are other transmitters. Instead, it is synthesized almost instantly as needed, and it then diffuses out of the presynaptic terminals over a period of seconds rather than being released in vesicular packets. Next, it diffuses into postsynaptic neurons nearby. In the postsynaptic neuron, it usually does not greatly alter the membrane potential but instead changes intracellular metabolic functions that modify neuronal excitability for seconds, minutes, or perhaps even longer. Then neuropeptides. Neuropeptides are synthesized differently and have actions that are usually slow and in other ways quite different from those of the small molecule transmitters. The neuropolypeptides are not synthesized in the cytosol of the presynaptic terminals. Instead, they are synthesized as integral parts of a large protein molecule by ribosome in the neuronal cell body. The protein molecule then enter the space inside the endoplasmic reticulum of the cell body and subsequently inside the Golgi apparatus, where two changes occur. First, the neuropeptide forming protein is enzymatically split into smaller fragments, some of which are the neuropeptide itself or a precursor of it. Second, the Golgi apparatus packages the neuropeptide into minute transmitter vesicles that are released into the cytoplasm. Then the transmitter vesicles are transported all the way to the tips of the nerve fibers by axonal streaming of the axon cytoplasm, traveling at the slow rate of only a few centimeters per day. Finally, these vesicles release their transmitter at the neuronal terminals in response to action potentials in the same manner as for small molecule transmitters. However, the vesicle is utilized and is not reused. Because of this laborious method of forming the neuropeptides, much smaller quantities of neuropeptides than of the small molecule transmitters are usually released. This difference is partly compensated for by the fact that neuropeptides are generally 1,000 or more times as potent as the small molecule transmitters. Another important characteristic of the neuropeptides is that they often cause much more prolonged actions. Some of these actions include prolonged closure of calcium channels, prolonged changes in the metabolic machinery of cells, prolonged changes in activation or deactivation of specific genes in the cell nucleus, and or prolonged alterations in number of excitatory or inhibitory receptors. Some of these effects last for days, but others last perhaps for months or years. Our knowledge of the function of neuropeptides is only beginning to develop. Next topic, effect of synaptic excitation on the postsynaptic membrane. Excitatory postsynaptic potential. So resting neuron is there. Then excited neuron undergoes excitation. Na plus influx will be happening. Negative 45 MV is created inside that neuron. And initial segment of axon will be there. Spread of action potential can be seen. Then inhibited neuron shows a negative 70 MV. Cl minus influx will be there. Then inhibitory K plus efflux will be happening. K plus travels out of the system from the resting neuron. So these are the three stages of a neuron. Resting neuron with a normal intraneuronal potential of negative 65 MV. Neuron in an excited state with a less negative intraneuronal potential caused by sodium influx, and neuron in an inhibited state with a more negative intraneuronal membrane potential, negative 70 MV, caused by potassium ion efflux, chloride ion influx, or both. So this shows resting neuron with an unexcited presynaptic terminal resting on its surface. The resting membrane potential everywhere in the soma is negative 65 MV. Then that shows presynaptic terminal that has secreted an excitatory transmitter into the cleft between the terminal and the neuronal somal membrane. This transmitter acts on the membrane excitatory receptor to increase the membrane's permeability to Na+. Because of the large sodium concentration gradient and large electrical negativity inside the neuron, sodium ions diffuse rapidly to the inside of the membrane. The rapid influx of positively charged sodium ions to the interior neutralizes part of the negativity of the resting membrane potential. The resting membrane potential has increased in the positive direction from negative 65 to negative 45 MV. This positive increase in voltage above the normal resting neuronal potential, that is to a less negative value, is called the excitatory postsynaptic potential, or EPSP. Excitatory postsynaptic potential, because if this potential rises high enough in the positive direction, it will elicit an action potential in the postsynaptic neuron, thus exciting it. In this case, the EPSP is positive 20 MV, that is 20 MV more positive than the resting value. Then discharge of a single presynaptic terminal can never increase the neuronal potential from negative 65 MV all the way up to negative 45 MV. An increase in this magnitude requires simultaneous discharge of many terminals, about 40 to 80 for the usual anterior motor neuron, at the same time or in rapid succession. This stimulus discharge occurs by a process called summation, which is discussed later. So, generation of action potentials in the initial segment of the axon, leaving the neuron threshold for excitation. When the EPSP rises high enough in the positive direction, there comes a point at which this initiates an action potential in the neuron. However, the action potential does not begin adjacent to the excitatory synapses. Instead, it begins in the initial segment of the axon, where the axon leaves the neuronal soma. The main reason for this point of origin of action potential is that the soma has relatively few voltage gated sodium channels in its membrane, and it makes it difficult for the EPSP to open the required number of sodium channels to elicit an action potential. Conversely, the membrane of the initial membrane segment has 7 times as great a concentration of voltage gated sodium channels as does the soma, and therefore can generate an action potential with much greater ease than can the soma. The EPSP that will elicit an action potential in the axon initial segment is between plus 10 and plus 20 MV, in contrast to the plus 30 or plus 40 MV or more required on the soma. Once the action potential begins, it travels peripherally along the axon and usually also backward over the soma. In some instances, it travels backward into the dendrites, but not into all of them, because they, like the neuronal soma, have very few voltage gated sodium channels and therefore frequently cannot generate action potentials at all. Then the threshold for excitation of the neuron is shown to be about negative 45 MV, which represents an EPSP of positive 20 MV, that is 20 MV more positive than the normal resting neuronal potential of negative 65 MV. Then electrical events during neuronal inhibition. Effect of inhibitory synapses on the postsynaptic membrane. Inhibitory postsynaptic potential. The inhibitory synapses open mainly chloride channels, allowing easy passage of chloride ions. To understand how the inhibitory synapses inhibit the postsynaptic neuron, we must recall what we learned about the Nernst potential for chloride ions. We calculated the Nernst potential for chloride ions to be about negative 70 MV. This potential is more negative than the negative 65 MV normally present inside the resting neuronal membrane. Therefore, opening the chloride channels will allow negatively charged chloride ions to move from the extracellular fluid to the interior, which will make the interior membrane potential more negative than normal, approaching the negative 70 MV level. Opening potassium channels will allow positively charged potassium ions to move to the exterior, and this will also make the interior membrane potential more negative than usual. Thus, both chloride influx and potassium efflux increase the degree of intracellular negativity, which is called hyperpolarization. This inhibits the neuron because the membrane potential is even more negative than the normal intracellular potential. Therefore, an increase in negativity beyond the normal resting membrane potential level is called an inhibitory postsynaptic potential, or IPSP. Effect of membrane potential caused by activation of inhibitory synapse, allowing chloride influx into the cell or potassium efflux out of the cell, which the membrane potential decreasing from its normal value of negative 65 MV to the more negative value of negative 70 MV. This membrane potential is 5 MV more negative than normal and is therefore an IPSP of negative 5 MV, which inhibits transmission of the nerve signal through the synapse. Then presynaptic inhibition. In addition to inhibition caused by inhibitory synapses operating at the neuronal membrane, which is called postsynaptic inhibition, another type of inhibition often occurs at the presynaptic terminals before the signal ever reaches the synapses. This type of inhibition is called presynaptic inhibition. So presynaptic inhibition is caused by release of an inhibitory substance onto the outside of presynaptic nerve fibers before their own ending terminate on the postsynaptic neuron. In most instances, the inhibitory transmitter substance is GABA. This release has a specific effect of opening anion channels, allowing large numbers of chloride ions to diffuse into the terminal fibril. The negative charges of these ions inhibit synaptic transmission because they cancel much of the excitatory effect of the positively charged sodium ions that also enter the terminal fibrils when an action potential arrives. Then presynaptic inhibition occurs in many of the sensory pathways in the nervous system. In fact, adjacent sensory nerve fibers often mutually inhibit one another, which minimizes sideways spread and mixing of signals in sensory tracts. Then, next topic, time course of the postsynaptic potentials. When an excitatory synapse excites the anterior motor neuron, the neuronal membrane becomes highly permeable to sodium ions for 1 to 2 milliseconds. During this very short time, enough sodium ions diffuse rapidly to the interior of the postsynaptic motor neuron to increase its intraneuronal potential by a few millivolts, thus creating the EPSP shown by the blue and green curves of figure, which won't be presented, but this potential has slowly declines over the next 15 milliseconds because this is the time required for the excess positive charges to leak out of the excited neuron and to re-establish the normal resting membrane potential. Precisely the opposite effect occurs for an IPSP, that is, the inhibitory synapse increases the permeability of the membrane to potassium or chloride ion or both for 1 to 2 milliseconds, and this action decreases the intraneuronal potential to a more negative value than normal, thereby creating the IPSP. This potential also dies away in about 15 milliseconds. Then other types of transmitter substances can excite or inhibit the postsynaptic neuron for much longer period, 400s of milliseconds or even 4 seconds, minutes or hours. This is especially true for some of the neuropeptic transmitters. Then some special characteristics of synaptic transmission. Fatigue of synaptic transmission. When excitatory synapses are repeatedly stimulated at a rapid rate, the number of discharges by the postsynaptic neuron is at first very great, but the firing rate becomes progressively less in exceeding milliseconds or seconds. This phenomenon is called fatigue of synaptic transmission. Fatigue is an exceedingly important characteristic of synaptic function because when areas of the nervous system becomes overexcited, fatigue causes them to lose this excess excitability after a while. For example, fatigue is probably the most important means by which the excess excitability of the brain during an epileptic seizure is finally subdued so that the seizure ceases. The development of fatigue is a protective mechanism against excess neuronal activity. So, summary of properties of a synapse, let's learn one by one. Unidirectional propagation of the nerve's impulse, we know that. Then delay. Subliminal fringe will be there. Temporal summation, occlusion, inhibitory or excitatory postsynaptic events. Inotropic fast transmission, then metabotropic slower transmission, neuromodulatory role, then fatigue. The mechanism of fatigue is mainly exhaustion or partial exhaustion of the stores of neurotransmitter substance in the presynaptic terminals. The excitatory terminals on many neurons can store enough excitatory transmitter to cause only about 10,000 action potentials, and the transmitter can be exhausted in only a few seconds to a few minutes of rapid stimulation. Part of the fatigue process probably results from two other factors as well, like progressive inactivation of many of the postsynaptic membrane receptors and slow development of abnormal concentrations of ions inside the postsynaptic neuronal cell. Then effect of acidosis or alkalosis on synaptic transmission. So most neurons are highly responsive to changes in pH of the surrounding interstitial fluid. Normally, alkalosis greatly increases neuronal excitability. For instance, a rise in arterial blood pH from the normal 7.4 to 7.8 to 8.0 often causes cerebral epileptic seizures because of increased excitability of some or all of the cerebral neurons. In a person who is predisposed to epileptic seizures, even a short period of hyperventilation, which blows off carbon dioxide and elevates the pH, may precipitate an epileptic attack. Then, conversely, acidosis greatly depresses neuronal activity. A fall in pH from 7.4 to below 7.0 usually causes a comatose state. For instance, in very severe diabetic or uremic acidosis, coma virtually almost always develops. Then, next topic, effect of hypoxia on synaptic transmission. Neuronal excitability is also highly dependent on an adequate supply of oxygen. Cessation of oxygen for only a few seconds can cause complete inexcitability of some neurons. This effect is observed when the brain's blood flow is temporarily interrupted because within 3 to 7 seconds the person becomes unconscious. Then some effective drugs on synaptic transmission. Many drugs are known to increase the excitability of neurons and others are known to decrease excitability. For instance, caffeine, theophylline, and theobromine, which are found in coffee, tea, and cocoa powder, respectively, like chocolate powder, all increase neuronal excitability, presumably by reducing the threshold for excitation of neurons. Strychnine is one of the best known of all agents that increase excitability of neurons. However, it does not do this by reducing the threshold for excitation of the neurons. Instead, it inhibits the action of some normally inhibitory transmitter substances, especially the inhibitory effect of glycine in the spinal cord. Therefore, the effects of the excitatory transmitters become overwhelming and the neurons become so excited that they go into rapidly repetitive discharge, resulting in severe tonic muscle spasms. Most anesthetic increase in neuronal membrane threshold for excitation and thereby decrease synaptic transmission at many points in the nerve system because many of the anesthetics are especially liquid soluble. It has been reasoned that some of them might change the physical characteristics of the neuronal membranes, making them less responsive to excitatory agents. Then next topic, synaptic delay. During transmission of a neuronal signal from a presynaptic neuron to postsynaptic neuron, a certain amount of time is consumed in the process of discharge of the transmitter substance by the presynaptic terminal, diffusion of the transmitter to the postsynaptic neuronal membrane, action of the transmitter on the membrane receptor, action of receptor to increase the membrane permeability, and inward diffusion of sodium to raise the EPSP to a high enough level to elicit an action potential. The minimal period of time required for all these events to take place, even when large numbers of excitatory synapses are stimulated simultaneously, is about 0.5 millisecond, which is called the synaptic decay. Neurophysiologists can measure the minimal delay time between an input volley of impulses into a pool of neurons and the consequent output volley. From the measure of delay time, one can then estimate the number of series neurons in the circuit. The next topic, convergence and divergence. Convergence occurs when multiple presynaptic neurons terminate on a single synapse. The presynaptic neurons can be inhibitory or excitatory, but offer a method of integrating several responses. Spatial summation, for example, requires convergence. Divergence occurs when one synapse has connections with two or more postsynaptic neurons, thereby offering a method to amplify the incoming signal to many downstream neurons. Then spatial and temporal summation. Spatial summation occurs when two or more separate presynaptic stimuli arrive at the synapse simultaneously. In this case, the individual postsynaptic potentials summate to give a threshold response. Then temporal summation occurs when the presynaptic neuronal stimuli arrive at the synapse rapidly one after the other in quick succession. The postsynaptic potential summates if the presynaptic stimuli are frequent enough. Then occlusion and subliminal fringe. The subliminal fringe occurs when a neuron receiving an excitatory input can affect synapses in its fringe or adjacent area. While this level of stimulation may not be enough on its own to generate a threshold response from the subliminal fringe neuron, a summated threshold response can be observed if the subliminal fringe neuron also receives excitatory inputs from other neighboring neurons simultaneously. In effect, this is a form of spatial summation leading to facilitation. If two neighboring neurons are excited separately, they have summed effects that are less than if they were excited at the same time. This is because these neighboring neurons can facilitate additional threshold responses from their overlapping fringe neurons. Neurons are said to be in the subliminal fringe if they are affected by excitatory input of an adjacent neuron but not brought to their fringe threshold. In a similar fashion, occlusion occurs when two adjacent neurons are given stronger excitatory inputs. This causes an overlap of excitation of the subliminal fringes of each neuron, such that the result of stimulating both adjacent neurons simultaneously would lead to lesser than expected response in contrast to the facilitation earlier. Clearly, the difference between the facilitation and occlusion is related to the strength of the incoming excitatory signal on two neurons that have an overlapping subliminal fringe area. When the incoming excitation is weak, summation can occur in the fringe neurons such that the response of simultaneous excitation of the two adjacent neurons is greater than their summed individual excitation. However, when the incoming excitation signal is stronger, overlap of excitation areas can occur such that a smaller than expected response is obtained when both adjacent neurons are stimulated simultaneously. Now let's learn about the next topic. That topic is about sensory receptors. So learning objectives include to classify the sensory receptors and give examples, then enumerate the properties of receptors, describe the labeled line principle and the law of projection. Then when we try to learn about the major terms associated with this topic, we could see terms like osmolality. We know that term, but what does that mean? A measure of the concentration or osmoles of solutes per kilogram of solvent. That is one osmolality. Then osmoreceptor, sensory receptor that detects changes in osmotic pressure in the blood, found in the hypothalamus and involved in regulating body fluid volume. Then transmembrane potential, difference in voltage between the interior and exterior of a cell. Our perceptions of signals within our bodies and of the world around us are mediated by a complex system of sensory receptors that detect such stimuli as touch, sound, light, pain, cold, and warmth. The purpose of this chapter is to discuss the basic mechanisms by which these receptors change sensory stimuli into nerve signals that are then conveyed to and processed in the central nervous system. So, types of sensory receptors and the stimuli they detect. There are many types of sensory receptors like mechanoreceptors, which detect mechanical compression or stretching of the receptor or of tissues adjacent to the receptor. Then thermoreceptors, which detect changes in temperature, with some receptors detecting cold and others warmth. Then nociceptors or pain receptors, which detect physical or chemical damage occurred in tissues. Then electromagnetic receptors. Osmoreceptor, sensory receptor that detects changes in osmotic pressure in the blood, found in the hypothalamus and involved in regulating body fluid volume. Then transmembrane potential, difference in voltage between the interior and exterior of a cell. Our perception of signals within our bodies of the world around us are mediated by a complex system of sensory receptors. So first we will be learning about the differential sensitivity of receptors. How do two types of sensory receptors detect different types of sensory stimuli? The answer is by differential sensitivities. That is, each type of receptor is highly sensitive to one type of stimulus for which it is designed, and yet is almost non-responsive to other types of sensory stimuli. Thus, the rods and cones of the eye are highly responsive to light, but are almost completely non-responsive to normal ranges of heat and cold, pressure on the eyeballs, or chemical changes in the blood. The osmoreceptors of the supraoptic nuclei in the hypothalamus detect minute changes in the osmolarity of the body fluids but have never been known to respond to sound. Finally, pain receptors in the skin are almost never stimulated by usually touch or pressure stimulus, but do become highly active the moment tactile stimuli become severe enough to damage the tissue. Then, modality of sensation, the labeled line principle. Each of the principal types of sensation that we can experience, like pain, touch, sight, sound, and so forth, is called a modality of sensation. Yet, despite the fact that we experience these different modalities of sensation, nerve fibers transmit only impulses. Therefore, how do different nerve fibers transmit different modalities of sensation? The answer is that each nerve tract terminates at a specific point in the central nervous system, and the type of sensation felt when a nerve fiber is stimulated is determined by the point in the nerve system to which the fiber leads. For instance, if a pain fiber is stimulated, the person perceiving pain, regardless of what type of stimulus excites the fiber, the stimulation can be electrically overheated, heating of the fiber, crushing of the fiber, or stimulation of the pain nerve ending by damage to the tissue cells. In all these instances, the person perceives pain. Likewise, if a touch fiber is stimulated by electrical excitation of a touch receptor or in any other way, the person perceives touch because touch fibers lead to specific touch areas in the brain. Similarly, fibers from the retina of the eye terminate in the vision areas of the brain. Fibers from the ear terminate in the auditory areas of the brain, and temperature fibers terminate in the temperate areas. The specificity of nerve fibers for transmitting only one modality of sensation is called the labeled line principle, and the impression of the specific stimulus arising from the specific nerve fiber is stimulated anywhere along its path to the brain is called the law of protection. So, this specificity of nerve fibers for transmitting only one modality of sensation is called labeled line principle, and the impression of the specific stimulus arising when the specific nerve fiber is stimulated anywhere along its path to the brain is called the law of protection. Projection. Next topic, modality of sensation. The labeled line principle. Each of the principal types of sensation that we can experience, pain, touch, sight, sound, and so forth, is called modality of sensation. Next topic, transduction of sensory stimuli into nerve impulses. Local electrical currents at nerve endings, receptor potentials. All sensory receptors have one feature in common. Whatever the type of stimulus that excites the receptor, its immediate effect is to change the membrane electrical potential of the receptor. This change in potential is called a receptor potential. So, mechanism of receptor potential, and we try to learn. Before that, classification of sensory receptors, let's try to learn. Mechanoreceptors, there are many different types of mechanoreceptors like skin tactile sensibilities like epidermis and dermis related, free nerve endings, expanded tip endings, Merkel discs plus several other variants, spray endings, Ruffini endings, encapsulated endings, Messner's corpuscles, then Krauss corpuscles, hair and organs, deep tissue sensibilities, free nerve endings, expanded tip. These are free tissue sensibilities like free nerve ending, expanded tip endings, spray endings, Ruffini endings, encapsulated endings, Pacinian corpuscles, plus a few other variants, muscle endings, muscle spindles, Golgi tendon receptors, then hearing related sensory receptors, sound receptors of cochlea, equilibrium related vascular receptors, then arterial pressure, baroreceptors of carotid sinus and aorta. Then thermoreceptors are of many kinds. Then cold receptors and warmth receptors are there. Then nociceptors are related to pain. Free nerve endings can be stated as an example of this. Then electromagnetic receptors, vision rods and cones. Chemoreceptors, taste related receptors of taste buds. Then smell receptors of olfactory epithelium. Arterial oxygen receptors of aortic and carotid bodies. Then osmolarity, neurons in or near supraoptic nuclei. Then blood CO2 receptors in or on surface of medulla and in aortic and carotid bodies. Blood glucose, amino acids, fatty acids, receptors in hypothalamus, etc. Then mechanisms of receptor potentials. Different receptors can be excited in one of the several ways to cause receptor potentials. By mechanical deformation of the receptor, which stretches the receptor membrane on opens ion channels. By application of chemical to the membrane, which also opens ion channels. By change of the temperature of the membrane, which alters the permeability of the membrane. And by the effects of electromagnetic radiation such as light on a retinal visual receptor, which either directly or indirectly changes the receptor membrane characteristics and allows ion to flow through membrane channels. These four means of exciting receptor correspond in general to the different types of known sensory receptors. In all instances, the basic cause of change in membrane potential is a change in membrane permeability of the receptor, which allows ions to diffuse more or less readily through the membrane and thereby to change the transmembrane potential. Several types of somatic sensory nerve endings, for example, free nerve endings, expanded tip receptor, tactile hair, Pacinian corpuscles, Messner's corpuscles, Krauss corpuscles, then Ruffini endings, Golgi tendon apparatus, some muscle spindles, etc. Next topic, maximum receptor potential amplitude. Maximum receptor potential amplitude. The maximum amplitude of most sensory receptor potentials is about 100 MV, but this level occurs only at an extremely high intensity of sensory stimulus. This is about the same maximum voltage recorded in action potentials and is also the change in voltage when the membrane becomes maximally permeable to sodium ions. Then relation of the receptor potential to action potentials. When the receptor potential rises above the threshold for eliciting action potentials in the nerve fiber attached to the receptor, action potentials occur. Then note also that more the receptor potential rises above the threshold level, the greater becomes the action potential frequency. Then receptor potential of the Pacinian corpuscle, an example of receptor function. Pacinian corpuscle has a central nerve fiber extending through its core. Surrounding the central nerve fiber are multiple concentric capsule layers, and this compression anywhere on the outside of capsule will elongate, indent, or otherwise deform the central fiber. Then shows only the central fiber of the Pacinian corpuscle after all capsule layers but one have been removed. The tip of the central fiber inside the capsule is unmyelinated. But the fiber does become myelinated. The blue sheath, no, it's not blue, but usually figure represent blue color. Then shortly before leaving the corpuscle to enter a peripheral sensory nerve. Then this shows that mechanism by which a receptor potential is produced in the Pacinian corpuscle. Observe the small area of the terminal fiber that has been deformed by compression of a capsule, and note that ion channels have opened in the membrane, allowing positively charged sodium ions to diffuse to the interior of the fiber. This action creates increased positivity inside the fiber, which is the receptor potential. The receptor potential in turn induces a local circuit of current flow, shown by the arrows, that spreads along the nerve fiber. At the first node of Ranvier, which lies inside the capsule of the Pacinian corpuscle, the local current flow depolarizes the fiber membrane at this node, which then sets off typical action potentials that are transmitted along the nerve fiber toward the central nerve system. Then relation between stimulus intensity and receptor potential. Changing amplitude of the receptor potential caused by progressively stronger mechanical compression, like increasing stimulus strength applied experimentally to the central core of a Pacinian corpuscle. The amplitude increased rapidly at first, but then progressively less rapidly at high stimulus strength. In turn, the frequency of repetitive action potentials terminated from sensory receptors increase approximately in proportion to the increase in receptor potential. Putting this principle together with the data, we could see that very intense stimulation of the receptor causes progressively less and less additional increase in numbers of action potentials. This exceedingly important principle is applicable to almost all sensory receptors. It allows the receptor to be sensitive to very weak sensory experience and yet not reach a maximum firing rate until the sensory experience is extreme. This feature allows the receptor to have an extreme range of response from very weak to very intense. Then adaptation of receptors. Another characteristic of all sensory receptors is that they adapt either partially or completely to any constant stimulus after a period of time. That is, when a continuous sensory stimulus is applied, the receptor responds at a high impulse rate at first and then at a progressively slower rate until finally the rate of action potentials decrease to very few or often none at all. The typical adaptation of certain types of receptors. Note that Pacinian corpuscles adapts very rapidly. Hair receptors adapt within a second or so, and some joint, capsule, and muscle spindle receptors adapt slowly. Furthermore, some sensory receptors adapt to far greater extent than do others. For example, the Pacinian corpuscle adapts to extinction within a few hundredths of a second, and the receptors of the bases of the hairs adapted to extinction within a second or more. It is probable that all other mechanoreceptors eventually adapt almost completely, but some require hours or days to do so. For that reason, they are called non-adapting receptors. The longest measured time for almost complete adaptation of a mechanoreceptor is about 2 days, which is the adaptation time for many carotid and aortic baroreceptors. However, some physiologists believe that these specialized baroreceptors never fully adapt. Some of the non-mechanoreceptors, the chemoreceptors and pain receptors, for instance, probably never adapt completely. Then nerve fibers that transmit different types of signals and their physiological classification. Some signals need to be transmitted to or from the central nervous system extremely rapidly. Otherwise, the information would be useless. An example of this is the sensory signals that apprise the brain of the momentary positions of the legs at each fraction of a second during running. At the other extreme, some types of sensory information, such as that depicting prolonged pain, do not need to be transmitted rapidly, and the slowly conducting fibers will suffice. The nerve fibers come in all sizes between 0.5 and 2.20 μm in diameter. The larger the diameter, the greater the conducting velocity. The range of conducting velocities is between 0.5 and 120 m per second. Then general classification of nerve fibers. General classification and sensory nerve classification of the different types of nerve fibers. In the general classification, the fibers are divided into types A, B, and type A fibers are then further divided into alpha, beta, gamma, and delta fibers. Type A fibers are the typical large and medium-sized myelinated fibers of spinal nerves. Type C fibers are the small unmyelinated nerve fibers that conduct impulses at low velocities. The C fibers constitute more than one half of the sensory fibers in most peripheral nerves, as well as all the postganglionic autonomic fibers. The sizes, velocities of conduction, and functions of the different nerve fiber types are also very important. Note that a few large myelinated fibers can transmit impulses at velocities as great as 120 m per second, covering a distance that is longer than a football field in 1 second. Conversely, the smallest fibers transmit impulses as slowly as 0.5 meter per second, requiring about 2 seconds to go from the big toe to the spinal cord. Alternate classification used by sensory physiologists. Certain recording techniques have made it possible to separate the type A alpha fibers into two subgroups, yet this same recording techniques cannot distinguish easily between A beta and A gamma fibers. Therefore, the following classification is frequently used by sensory physiologists. That is group 1A. Fibers from the annulospiral endings of muscle spindles, about 17 mm in diameter on average. These fibers are alpha type A fibers in the general classification. Then group 1B, these are fibers from the Golgi tendon organs, about 16 mm in diameter on average. These fibers also are alpha type A proteins fibers. Then group 2, fibers from most discrete cutaneous tactile receptors and from the flower spray endings of the muscle spindles, about 8 mm in diameter on average. The fibers are beta and gamma type, a fiber in the general classification. Then group 3, fibers carrying temperature, crude touch, and pricking pain sensations, about 3 mm in diameter on average. They are alpha type of A fibers in the general classification. Then group 4, unmyelinated fibers carrying pain, itch, temperature, and crude touch sensations, 0.5 to 2 mm in diameter. They are type C fibers in the general classification. Then transmission of signals of different intensity nerve tracts, spatial and temporal summation. One of the characteristics of each signal that always must be conveyed is signal intensity. For instance, the intensity of pain. The different gradations of intensity can be transmitted either by using increasing numbers of parallel fibers or by sending more action potentials along a single fiber. These two mechanisms are called respectively spatial summation and temporal summation. When we tried to learn about this, we could see pattern of stimulation of pain fibers in a nerve leading from an area of skin pricked by a pin. So we can see weak stimulus, moderate stimulus, strong stimulus. Then spatial summation, whereby increasing signal strength is transmitted by using progressively greater numbers of fibers. This figure shows a section of skin innervated by a large number of parallel pin fibers. Such of the fibers arborizes into hundreds of minute free nerve endings that serve as pain receptors. The entire cluster of fibers from one pain fiber frequently covers an area of skin as large as 5 cm in diameter. This area is called the receptor field of that fiber. The number of endings is large in the center of field but diminishes toward the periphery. One can also see from the figure that arborizing fibrils overlap those from other pain fibers. Therefore, a pinprick of the skin usually stimulates endings from many different pain fibers simultaneously. When the pinprick is in the center of the receptive field of a particular pain fiber, the degree of stimulation of that fiber is far greater than when it is in the periphery of the field, because the greater the free nerve endings in the middle of the field is much greater than at the periphery. Thus, the lower part shows three views of cross sections of nerve bundle leading from the skin area. To the left is the effect of weak stimulus with only a single nerve fiber in the middle of the bundle stimulated strongly. Then, whereas several adjacent fibers are stimulated weakly. The other two views, the nerve cross section allows the effect of moderate stimulus and strong stimulus, and progressively more fibers being stimulated. Thus, the strong signals spread to more and more fibers. This process is the phenomenon of spatial summation. Next topic, temporal summation. A second means of transmitting signals of increasing strength is by increasing the frequency of nerve impulsion in each fiber, which is called temporal summation. That demonstrates the phenomenon showing an upper part of a changing strength of a signal and in the lower part an actual impulse transmitted by the nerve fiber. When we try to learn the summary of properties of receptors, differential sensitivities will be there. Ordered projection to the cortex labeled line, then graded responses adaptable. These are the properties of receptors. Next major topic, somatic sensory pathways. Learning objectives include describing the tactile receptors, then describe the Weber Fechner law and power law. I'm not sure about the pronunciation. Then enumerate the characteristics of sensory transmission. Trace the ascending pathways and describe the sensations carried by them. Now let's learn major terms associated with this topic. Anterolateral system, a tract carrying sensory signals upward, which immediately cross to the opposite side of the code and ascend through the anterior and lateral white columns of the code. Then dorsal column medial lemniscal system, a tract carrying sensory signals upward, mainly in the dorsal columns of the code. The tract crosses to the opposite side of the medulla and continues upward through the brainstem to the thalamus by way of medial lemniscus. Then proprioception, revealing the sense of position. Somatic, related to the body. Tactile, relating to the sense of touch. The somatic senses are the nerves mechanism that collect sensory information from all over the body. The senses are in contradiction to the special senses, which mean specifically vision, hearing, smell, taste, and equilibrium. The attributes of sensations are their equality or types as described later, intensity, duration, and localizations. Then let's learn about classifications of somatic senses. The somatic senses can be classified into three physiological types. The mechanoreceptive somatic senses, which include both tactile and position sensations that are stimulated by mechanical displacement of some tissues of the body. The thermoreceptive senses, which detect heat and cold. The pain sense, which is activated by factors that damage the tissue. This chapter deals with the mechanoreceptive, tactile, and position senses. So, first, other classifications of somatic sensations. Somatic sensations are also often grouped together in other classes as follows. Exteroreceptive sensations are those from the surface of the body. Proprioceptive sensations are those relating to the physical state of the body, including position sensations, tendon and muscle sensations, pressure sensations from the bottom of the feet, and even the sensations of equilibrium, which is often considered a special sensation rather than a somatic sensation. Visceral sensations are those from the viscera of the body. In using this term, one usually refers specifically to the sensations from the internal organs. Then deep sensations are those that come from deep tissues, such as from muscles and bones. The sensations include mainly deep pressure, pain, and vibration. Then detection and transmission of tactile sensations. Interrelations among the tactile sensations of touch, pressure, and vibration. Although touch, pressure, and vibration are frequently classified as separate sensations, they are all detected by the same types of receptors. There are three principal differences among them. Touch sensation generally results from stimulation of tactile receptors in the skin or in tissues immediately beneath the skin. Then, pressure sensation generally resulting from deformation of deeper tissues. Vibration sensation resulting from rapidly repetitive sensory signals, but some of the same types of receptors as those for touch and pressure are used. Then, tactile receptors. There are at least 6 entirely different types of tactile receptors, but many more similar to these also exist. First, some free nerve endings, which are found everywhere in the skin and in many other tissues, can detect touch and pressure. For instance, even light contact with the cornea of the eye, which contains no other type of nerve ending besides free nerve endings, can nevertheless elicit touch and pressure sensations. Second, touch receptor with great sensitivity is the Meissner corpuscle, an elongated encapsulated nerve ending of a large myelinated sensory nerve fiber. Inside the capsulation are many branching terminal nerve filaments. These corpuscles are present in the non-hairy parts of the skin and are particularly abundant in the fingertips, lips, and other areas of the skin where one's ability to discern spatial locations of touch sensations is highly developed. Meissner corpuscles adapt in fraction of a second after they are stimulated, which means that they are particularly sensitive to movement of objects over the surface of skin as well as to low frequency vibration. Then third, the fingertips and other areas that contain large numbers of Meissner corpuscles usually also contain large numbers of expanded tip tactile receptors, one type of which is Merkel discs. Then hairy parts of the skin also contain moderate numbers of expanded tip receptors, even though they have almost no Meissner corpuscles. These receptors differ from Meissner corpuscle in that they transmit an initially strong but partially adapting signal and then a continuing weaker signal that adopts only slowly. Therefore, they are responsible for giving steady state signals that allow one to determine continuous touch of objects against the skin. Merkel discs are often grouped together in a receptor organ called the lagodome receptor, which projects upward against the underside of the epithelium of the skin, as is also shown, and this upward projection causes the epithelium at this point to protrude upward, thus creating a dome and constructing an extremely sensitive receptor. Also note that the entire group of Merkel discs is innervated by a single large myelinated nerve fiber. These receptors, along with the Meissner corpuscle discussed earlier, play extremely important roles in localizing touch sensations to specific surface areas of the body and in determining the texture of what is felt. Fourth, slight movement of any hair on the body stimulates a nerve fiber twining its base. Thus, each hair and its basal nerve fibers, called the hair end organ, are also touch receptors. A receptor adapts rapidly and, like Meissner corpuscle, detects mainly the movement of objects on the surface of body or initial contact with the body. Fifth, located in the deeper layers of the skin and also in still deeper internal tissues are many Ruffini endings, which are multi-branched encapsulated endings. These endings adapt very slowly and therefore are important for signaling continuous states of deformation of the tissue, such as heavy prolonged touch and pressure signals. They are also found in joint capsules and help to signal the degree of joint rotation. Then sixth, Pacinian corpuscles, which are discussed earlier, lie both immediately beneath the skin and deep in the facial tissues of the body. They are stimulated by rapid local compression of the tissues because they adapt in a few hundredths of a second. Therefore, they are particularly important for detecting tissue vibration or other rapid changes in the mechanical state of the tissue. So that's all about this topic. Tomorrow we will be continuing this portion. Thank you for listening.